Welcome to our presentation. We are proud to introduce our new biomaterial as a care and cure product specialized for people who are interested in chronological aging. In this presentation, we will discuss what exactly is OligoM, the know-how behind OligoM, our current patents, and also the story of why we need OligoM. First of all, what is OligoM? Well, let's look at it this way. If you take a look at the Chinese characters for crisis, you can see that one meaning is danger, but the other meaning is opportunity. We can see danger, but from a new viewpoint, we can see the opportunity to prevent a crisis. Let's take a look at some of the current dangers facing society, like aging, menopause, osteoporosis, and obesity. We can see all of these as common dangers related to metabolic syndrome. But if we switch our viewpoint of crisis into one of opportunity, we can see natural food products from agriculture and aquaculture that can be used to fight metabolic syndrome like alginate oligosaccharides, lectins, quercetin, and curcumin. We chose to focus on alginate oligosaccharide. Here, we will discuss the role of alginate oligosaccharide originated from edible seaweed. The key to studying the functional activity of alginate oligosaccharide is through first gathering large-scale data via three types of omics technology: transcriptomics, proteomics, and metabolomics. After gathering the omics data, the data was profiled by, first, the grouping of the molecules into a dataset based on expression levels. Second, the identification of significant molecules through enrichment analysis. Third, performing a functional analysis of all significant molecules. Fourth, finding putative upstream regulators. And finally, by network prediction analysis. What exactly is alginate oligosaccharide? After the ingestion of a polymer of seaweed, two types of alginate oligosaccharide, consisting of isomeric monomers of guluronic acid and manuronic acid, are produced by the intestinal microorganisms in sea urchins and abalone. The two types of alginate oligosaccharide are called OligoG and OligoM. OligoG consists mainly of linked guluronic acid, whereas OligoM consists mainly of linked manuronic acid. OligoG by Algae Pharma has already been granted orphan drug designation by the FDA as of February 2016 in treating cystic fibrosis. It has successfully passed through Phase 2 trials in the UK and Ireland. As a drug, OligoG is highly water-soluble, stable, non-toxic, and biocompatible. Its benefits have been demonstrated in vitro and in vivo studies. As an isomer of OligoG with similar properties, OligoM has a high potential in the future as a candidate drug independent from OligoG. Although OligoG and OligoM are structural isomers with the same molecular weight, they differ in structure quite significantly. As you can see for OligoM at the top, M's are linked together in a linear conformation. But for OligoG at the bottom, G's are linked in an angular conformation. With different conformations, the two alginate oligosaccharides also have different biological functions. For example, when it comes to the activation of phosphorylated AMPK, a substance that is related to preventing diabetes and obesity when upregulated, M3 and M5 of OligoM are strong AMPK activators. However, G3 and G5 of OligoG are weaker AMPK activators in comparison. Therefore, OligoM is a strong candidate as a bioactive material for treating metabolic syndromes like obesity and diabetes. Next, let's look at the know-how of our product, our specialization for making OligoM. First of all, we have a patent for the enzyme and non-GMO strain required for making OligoM, along with full sequencing and proteomic information. We have also completed all bioactive material composition analysis of OligoM using mass spectrometry. Finally, through pilot testing, we have optimized the full process for making OligoM from raw materials into two types of products, liquid or powder, both with high purity and high yields. There are two methods for making OligoM: acid hydrolysis and enzyme hydrolysis. As shown here, acid hydrolysis produces low amounts of unsaturated oligomers, whereas enzyme hydrolysis, the process that we use for making OligoM, produces high amounts of unsaturated oligomers with higher activity. Also, acid hydrolysis yields higher impurities than enzyme hydrolysis. In terms of M to G ratio, our OligoM contains 91.9% manuronate and 8.1% glucuronate at a ratio of 11.3, while acid hydrolyzed alginate oligosaccharide contains 56% manuronate and 44% glucuronate at a ratio of 1.19. Next, we will discuss our patents related to OligoM. This is the list of our OligoM-related patents. Our currently registered patents involve the effects of OligoM on microflora changes and the effects of OligoM on estrogen-like activity in the menopausal signaling pathway. Next, here is the story of why we need OligoM. This is a map of average BMIs in different countries across the globe. A high BMI indicates high risks for obesity and is also related to an increase in menopausal symptoms. Two related topics that can be targeted using OligoM in response to high BMIs are anti-aging and sports nutrition. For checking the anti-aging effects of OligoM, and in order for it to be approved as a functional food or sports drink, a toxicity evaluation was performed using the listed testing methods. We also verified estrogen-like activity, completed an evaluation of bone mineral density, and investigated mechanisms of osteoporosis using an old mouse in vivo model. The next step is human trials, and we are currently looking for partners with whom to perform human trials using OligoM. Our material shows high potential for OligoM as a new drug candidate. So we are interested in developing new business partnerships as well as further R&D collaboration. In our chronological aging research, we tested the effects of 0.2 milligrams per milliliter of OligoM on the intestinal microflora of 17th-month-old mice, the equivalent of 80-year-old humans, for 10 weeks. After the feeding experiment, we found that the microflora changed from old to young into similar to that of 1-month-old mice. When looking at the principal coordinates analysis plot, we can see that the 17th-month-old OligoM mice had a similar pattern as the 1-month-old control mice. As for the relative abundance of microbial phylotypes, there were 3 microbes of particular interest that are related to osteoporosis: Bacterioides acidophasians, Prevotella species, and Lactobacillus species. In the old mouse control group, there was low abundance of these 3 microflora, but treatment with OligoM allowed for higher relative abundance. Here, we have reported for the first time that OligoM has estrogen-like effectiveness and that it could have an impact on menopause. OligoM activates PGC1-alpha to express ERR-alpha, which then activates estrogen-dependent target genes. By looking into the effect of OligoM and estradiol on the ER-dependent genes, PS2 and PR in MCF7 cells, OligoM increased their relative expressions and worked synergistically with estradiol when treated together. CTSD, on the other hand, didn't increase when combined with estradiol, which is good because its expression is related with a cancer signaling pathway. We can see that OligoM and estradiol regulate ER-dependent genes in a positive manner. These 4 genes were highly induced in OligoM treatment, which proves that OligoM acts on ER-dependent gene expression without the presence of estrogen. We also tested the effects of OligoM on osteoporosis by measuring the bone mineral density of aging 17-month-old mice. After 12 weeks of feeding, we measured the density using computed tomography, CT scans. At 4 weeks of feeding, we further split the mice into exercise groups and no exercise groups. As you can see in the femur, or upper bone in the leg, there was a 7.1% increase in bone mineral density of the OligoM group in comparison to the sterilized distilled water group. When exercise is also included as a factor, there is a 6.6% increase in bone density for the OligoM group in comparison to the water and exercise group. In total comparison to the sterilized distilled water group with no exercise, the OligoM exercise group increased bone mineral density by 27.7% in 20-month-old mice. So, exercise and OligoM together help to treat osteoporosis better than just exercise or just water. We also measured the bone mineral density in the tibia. As you can see, OligoM increased bone mineral density by 8.6% in comparison to water and by 15.1% when exercise was included. OligoM and exercise combined increased the bone mineral density by 50.8% when compared to water and no exercise treatment in 20-month-old mice. Here are the CT scan images that compare sterilized distilled water treatment and OligoM with exercise treatment. The images show a clear increase in bone density accumulation in the OligoM and exercise-treated mice. Here is the osteoporosis signaling network that shows the proteome after OligoM treatment in comparison to the osteoporosis proteome database. It is highly linked and very complex. However, it becomes more simplified when looking at the Osteoblast differentiation pathway. After treatment with OligoM, OligoM activates OPG and osteocalcin, but it blocks the expression of TNF and IL-1, an osteoclast-activating cytokine. Let's move on to application in sports nutrition. Here you can see the market of sports nutrition. In 2011, the U.S. supplement market reached $30 billion, more than $3 billion of that being from sports nutrition, and it is expected to rise in 2019 to $38 billion, possibly around $4.5 billion US for sports nutrition. Here is a chart comparing the effects of exercise for 16 weeks on the body weight of aging mice in control and OligoM treatment groups. After exercise treatment of the aging mice, we measured their body weights. While there was no significant change in the water control group, there was a decrease in weight for the OligoM group. We also compared how far the chronologically aging mice could run before getting exhausted. After 16 weeks, there was a 2-kilometer difference between the two groups. The OligoM group could run 2 kilometers more per week than the plain water group. Thus, OligoM shows to have a high impact on the exercise ability of chronological aging mice. In order to biologically explain the effects of OligoM on the exercise ability of aging mice and its future in sports nutrition, we were able to conduct in vitro, in vivo, and pre-trials in humans. In our in vitro study, we found that OligoM is a strong phosphorylated AMPK activator and a strong phosphorylated ACC activator, substances that allow for improved exercise ability. In our in vivo study using high-fat diet-induced mouse models, we found that OligoM is related with TCA cycle activation and fatty acid oxidation to allow for the generation of ATP in a short period of time. In our pre-trials for future human trials, we saw the effects of OligoM and exercise on aerobic performance. OligoM increased the cycling speed and power of subjects who drank OligoM 2 times using the Wingate experiment. From this pre-testing, we saw that after heavy exercise, there was no evidence of muscle pain caused by lactic acid accumulation in people who took OligoM. Thus, we discovered that OligoM activated gluconeogenesis to convert lactic acid in muscle into glucose in the liver. We are currently collaborating with Texas A&M University's Department of Health and Kinesiology for functional food and development using OligoM. And we hope to further collaborate with other international business trading partners. If you have any questions or are interested in intellectual property rights transfer or R&D collaboration, please contact us at dwkim@jnu.ac.kr or dwkim@tamu.edu. Thank you for watching our presentation.